Hello and welcome to Nikolay's genetics lessons, and today's video going to be about population genetics. And here's a problem. Consider a population 70% homozygous dominant, 28% heterozygous, and 2% homozygous recessive. Reasoning from genotypic frequencies, does this population appear to be at Hardy-Weinberg equilibrium or not? So here is 4 answers to choose from. Answer A. Yes, the genotypic frequencies are very close to those expected. Answer B. No, there are way too many homozygous dominant organisms. Answer C. No, there are way too many heterozygous organisms. And answer D. No, there are way too few homozygous recessive organisms. If you think that you can solve this problem, you can pause video here, try to solve this problem on your own first, and when you would be ready, you can run video again and you can compare your answers with my answers and explanations. So here is how we are going to solve this problem. First of all, we have to know Hardy-Weinberg equilibrium, or formula that describes this equilibrium, and here is a formula. So frequencies of the 3 genotypes in gene pool would equal to p squared plus 2pq plus q squared, and when we add all the 3 genotypic frequencies, we are going to get 1 or 100%. As you see, we have here 2 alleles, allele p and allele q. If we add these frequencies, we should get 1. So 2 alleles, P and Q. In our case, this is allele E, so P stands for the dominant allele E and Q stands for the recessive allele E. When we add these 2 alleles in the gene pool, of course we are going to get 100%. And these 2 alleles can make 3 genotypes. So P squared in this formula stands for the homozygous dominant genotype. 2pq stands for the heterozygous genotype, so capital E, small e, and q squared stands for the homozygous recessive genotype, so small e, small e. So 2 alleles can make 3 genotypes. So far it is very easy and understandable. So what we know about the frequencies? We know that the frequency of the homozygous dominant genotype equals to 70%, and that means that on the scale between 0 and 1, and as you see we have here the biggest number that equals to 1, we would have 0.7 or 70, that is the same. So it is very easy to convert one frequencies into the other frequencies. We just have to remember that on the scale between 0 and 1, 100% equals to 1 on the scale between 0 and 100%. So if we have, for example, 70%, that means 0.7. So if we have frequency of the heterozygous organism 28% on the scale between 0 and 1, this is going to be 0.28. And homozygous recessive, that is 2%, would be 0.02. When we add all these 3 frequencies, as you see, we got 1 or 100%. So far our calculations are good. So in order to find if these frequencies are in balance, and what does it mean? That means that with each following generation we are going to get the same frequencies of these 3 organisms, so the 3 genotypes would be in balance and frequencies would be stable. So in order to do these calculations, so you have to understand that these frequencies are observed frequencies. So we can put OB here, observed frequencies. And now we have to calculate frequencies, that is expected frequencies. So how we can do it? We have to find number P and number Q, or we have to find frequency of the dominant allele and recessive allele, and then do our calculations according to this formula. So what is the frequency of the dominant allele? E or P, according to the formula here. And we have, as you see, two dominant alleles here, and here we have, as you see, heterozygous organism that is present in the frequency 0.28, and as you see, each heterozygous organism have dominant allele and recessive allele. So half of this number we have to add here, and this is going to be 0.14, and half of this alleles that is present here are actually recessive allele E, so we have to add this number here, also 0.14, and now we know the frequency of the dominant allele, that is E, and this equals to 0.84, and frequency of the recessive allele E would equal to 0.16. So when we add these two frequencies, as you see, we are going to get 1 or 100%. Once again, this is observed numbers of 3 genotypes frequencies, and this is frequency of each allele in this gene pool. Now we have to calculate expected frequency in this gene pool. So according to this formula, P squared stands for the homozygous dominant genotype, so P squared would equal to frequency of the dominant allele P squared, so 0.84 multiplied by itself or squared, and frequency would equal to 0.71. I'm rounding numbers to 2 decimal places, and according to this formula, number of heterozygous organism or frequency of this genotype would equal to 2pq. We know number p, that equals to 0.84, and we know the frequency of the allele q or recessive allele e, that equals to 0.16. If we multiply these numbers, we are going to get 0.27, and the last calculations would be, we have to find frequency of the homozygous recessive genotype, expected frequency, so this equals to q squared, and we know the frequency of the recessive allele E in this gene pool, that equals to 0.16, so we have to square this number, and we are going to get here 0.0256. As you see, this is close to 0.02. So if we add all these frequencies, once again, we are going to get number that is very close to 100%. And as you see, frequencies also are very close. So number of observed heterozygous organisms equals to 28%, and we see here expected number is 27, and here we have 0.02 or 2%, and here we have 2.5%. The number that is very close to this number also. So what we can say that numbers expected are very close to numbers or frequencies that is observed frequencies. So our answer would be answer A. Yes, the genotypic frequencies are very close to those expected. So in other words, genotypically this community is very stable, and numbers from one generation to the other generation are almost the same. So that means that there is almost no external influences like migration that may influence frequencies of these alleles in the gene pool, and probably there is no assortative mating that may also affect frequencies of the alleles in the gene pool, and probably all three genotypes has the same fitness. So the small deviation of the expected ratios from the observed ratios or frequencies probably can be explained by the slow mutation rate, and this is something that is unavoidable in any community. So, as you see, the correct answer would be answer A. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.